And without further ado, I would like to introduce today's speaker, Dr. Howard Fields. He received his MD and PhD in neuroscience at Stanford in 1965 and 1966. Following clinical training in neurology at Harvard Medical School in 1972, he joined the faculty of UCSF, where he's currently Professor of Neurology and Physiology Emeritus. He was founder of the UCSF Pain Management Center and made major contributions to understanding and treating post-herpetic neuralgia, including demonstrating the effectiveness of opioids and topical lidocaine. In laboratory studies with Alan Basbaum, he discovered and elucidated a pain-modulating neural circuit that engages endogenous opioids and is activated by opioid analgesics. Furthermore, with John Levine, he discovered that placebo analgesia can be blocked by an opioid antagonist. He received numerous awards, including the National Migraine Foundation Award, the Bristol Myers Award for Pain Research, a Merit Award from the NIH, the CARE Award from the American Pain Society, and the Kotzias Award from the American Academy of Neurology. He's given numerous distinguished lectures at Harvard Medical School, and in 1997, he was elected to the membership of the Institute of Medicine, in 2010 to the American Academy of Arts and Sciences. In 2018 and 19, he served as a member of the congressionally mandated Pain Management Task Force to develop guidelines for the treatment of chronic pain. I can't say enough how much of a luminary Howard Fields is. He's also a personal friend, and I'm just so pleased and honored that he's here to join us today and share with us results of his research and also share his wisdom. He's here to speak with us today about self-fulfilling prophecies, how predictions and expectations modulate pain. Howard, the floor is yours.
Okay, great. It's wonderful to be here. And thank you, Beth, for inviting me. And thanks, Ashley, so much for doing the logistics. This seems to be working out pretty well. I always love to give a talk at Stanford. It's kind of a second home for me. The pain group at Stanford is just, in my mind, it's a model for what all places should be doing. So today, what I'm hoping I'll do is change your mind a little bit about how your brain processes a pain signal. I also hope to convince you that understanding how these changes in the central nervous system in the setting of chronic pain are critical to understanding your patients, and hopefully will help you to take care of them a little better. Okay, I have some disclosures that I want to make. First, that I'm an expert witness defending Johnson & Johnson in opioid litigation. The other conflict is that I'm a neurologist, so I tend to believe that what's going on in the brain is probably the most important thing in medicine. I also would like to dedicate this talk today to Don Kennedy, who died a couple of years ago in the early COVID epidemic. He was my PhD mentor, and he was head of the Food and Drug Administration. He also was president of Stanford University at a critical time for the university, and just a dear person that I miss and love. I graduated from Stanford Medical School in June of 1965. Though the days that I spent there were among the happiest of my life. So today I'm going to talk a little bit about pain, some of the circuitry that's involved, and how that might change and how that might differ in patients with chronic pain. So this is the canonical pain model that we all pretty much accept, and I think it's very much validated. The idea is that you activate nociceptive terminals in the peripheral tissues, musculoskeletal, right? The skin, the brain, you name it, blood vessels, they're all innervated by nociceptive fibers. When there's impending tissue damage or any kind of intense stimulus, these fibers, they tend to be of small diameter, slowly conducting, are activated. They transmit that message to the superficial layers of the dorsal horn. Their neurotransmitter is glutamate. They activate second and third order cells. These cells then send their axons to the opposite ventral part of the spinal cord. The spinothalamic tract is the major one that we know about, but there are spinoreticular, spinoparabrachial projections as well that were later discovered. Once these terminals get to the thalamus, the signal is distributed to a variety of cortical areas. And this was only understood after we developed functional magnetic resonance imaging, which allowed us to correlate activity in specific parts of the brain with an awake patient telling you the level of pain intensity that they were experiencing. And what's shown on this side, these areas that are sort of light green and red, are areas that show a correlation, a highly significant correlation with patients' reported pain intensity. So this is the anterior cingulate, somatosensory cortex, the insula, and the second somatosensory cortex. So presumably, when those areas are activated, then you have this mysterious subjective perceptual event, which we call pain, right? So I'm a neurologist, and for me, pain is the activity in this circuit that leads to people telling you that they have pain. But chronic pain is quite different. And I'm sure that many of you in the audience, and I know Beth for sure, understands that these patients, the chronic pain is different in a lot of ways. One of the most important ways is that the reported pain intensity is not consistently correlated, or it's not correlated in a lawful way with the level of tissue damage. And that's very different from acute pain, where you can actually adjust the intensity of the stimulus to point where you get to potential tissue damage, and then people will tell you, yeah, that's pain, that's more painful, that's more painful, and they can rate it 0 to 10. There's nothing there. It's very difficult to correlate any kind of pathology that you can see with imaging or on physical exam with the level of pain intensity that people report. And I hope that by the end of this lecture, you'll understand that a little bit better. Frequently, there's psychological comorbidity. This is something we often don't like to discuss with patients, but it's pretty clear that under certain circumstances, anxiety and depression can be correlated with a worsening of pain intensity. We'd like to know how that comes about. But what I mostly want to talk about today is how learning and expectation, and this will be normal learning, but with a persistent input of noxious stimuli or some persistent peripheral tissue process that leads to learning and expectation. That changes the wiring in the brain and changes the way people experience it. So I'd like to talk about a study that I did in collaboration with colleagues at Northwestern University, Marwan Baliki and Vanya Abkarian. And in this study, we compared normal subjects with age match patients with chronic back pain. It was a relatively small study, just about 8 people in each group. And what we're looking at here is just an applied stimulus. We're not looking at the chronic pain process that's going on. That's been going on for years in the pain patients. But what we're doing here is comparing how a pain signal is processed in people with chronic pain compared to those who are normal. So what you see up here is each one of these little boxes is the application of a pain stimulus of varying intensity and varying duration. And above here is the pain report. You can see that as the pain's applied, the report of pain tracks it pretty well in time. Below is processed artificial color showing areas of the brain that are activated by this acute painful stimulus. And what's shown in this track here, the top level of images, is the parts of the brain that are similar in terms of being activated by these stimuli in the normals and the chronic back pain. Down below is a subtraction. So what areas show a difference between chronic pain and normals in the response of the nervous system to an acute noxious stimulus? And there turns out that what we found was the main place where this happened, where there was a very significant, statistically significant difference, was in an area down here. Let me blow that up. And this is an area called the ventral striatum, or the nucleus accumbens, right? So this is a part of the brain that's going to come up again and again. It's not traditionally thought of as being part of the pain processing system, but it's consistently activated. And this is the one area that really does seem to be markedly different in people with chronic low back pain compared to normals. And this is in how they handle a typical acute noxious stimulus. This was thermal pain. Okay, so hopefully you're scratching your head and saying, well, what's going on here? Does this change make any sense biologically, right? Why would you want to change how you process a nociceptive signal over time? That there has to be an explanation for that. And then, some of you might actually realize that this nucleus accumbens ventral striatum area is part of the so-called reward circuit. I think people are starting to change their mind now about that region of the brain and thinking, well, maybe it isn't just pain, maybe it's involved in all types of value assessment. Okay. So when I got to Stanford Medical School, Don Kennedy had just been hired, and he was hired as an assistant professor in the biology department, and he gave a course on animal behavior. I was in my second year at Stanford Medical School, and I took his course, and so I in my research career, I have to say that I started off in a biology department. So evolution is a big part of what you learn in biology. And so here's a few principles that I think are important to keep in mind. One is, we often think about, well, here's our brain, here's our self, there's our mind. Somebody comes and, whacks us on the arm, with a hammer, and hurts. The signal is transmitted, comes to the brain. It feels like it's a passive event, right? But if you think about it for a moment from an evolutionary perspective, any kind of sensory information is useless unless you can act on it, right? So during this lecture, what I'm going to try to convince you of is the only way you can fully understand pain is in the context of how that information is used to guide behavior, right? It is, so understanding the pain sensory system is not enough. You have to understand how the brain uses that information to benefit the individual, right? So to me, this is sort of the key principle that I'm trying to get across, and it's quite simple. There are two distinct biological functions of pain, and neither one of them have to do with the subjective experience that brings people to see a physician. One is it produces a motivation, right? The motivation is, I got to stop this thing. I either have to escape, get away. I have to do something to minimize tissue damage. So that's the biological. That's sort of the immediate biological effect of the activity in your pain sensory system. Okay, motivation, right? So motivation, what is motivation? It's what drives action, right? So again, gets back to that point that I was making before. Sensation is meaningless unless you can act on it. And pain is intrinsically motivational. It's that you can't have pain without this motivation to escape. But the other point, and the one that I'm going to mostly focus on today, is that pain is a teaching signal, right? You remember things that are painful and you avoid those situations. You're more careful in the future. You know, if you slam the car door and you got your thumb caught, next time you hold your arm closer to your body when you're closing the door, right? This is function number 2. It's a teaching signal to inform future decisions, again, to minimize tissue damage. 1, immediate, 2, future situations. Okay, so here's a nice test that we used in the laboratory to assess pain. It's a 3-compartment box. You let the rat roam around in it, and then you close it off. There's a little guillotine door that slides shut. You confine the animal. We were working with rats to one of the compartments, and as you can see below, there's a little camera. You can apply a noxious stimulus through the floor with a thermal stimulator. And if you do that, hold the animal in there, say for a few minutes, deliver a noxious stimulus, and then let the animal run around, the animal will avoid the room in which it's experienced a noxious stimulus. So, for example, one typical model that's used in pain research is injecting a dilute concentration of formalin. It produces an immediate pain that lasts for about an hour and a half. Then the animals are okay. But then you test them and they learn to avoid where they've gotten that noxious stimulus, right? Okay, so what's going on here? The teaching signal alters predictions that govern actions. That's what we've just seen in this behavioral test that I just illustrated. So you're in a situation, it's a given context, you've been there before, there are a bunch of cues going on. And based on that, in your brain, you make a prediction. Oh my God, you know, the last time I was in there, something bad happened, it hurt, and I really got to get out of there. So that's the action. And then there'll be an outcome, right? Now, if the outcome is better than you expected, let's say that you got out of there, you avoided the pain, or you had ongoing pain, you engaged in action, and that reduced the pain, right? Or you made a prediction, maybe it was a bad prediction, you had an action, you went to the other room, and let's say that there was some food in the other room. You learn, that's a reward, that's what's called positive reinforcement. And then that information is fed back. The next time you're in that situation, your predictions have changed, right? So now the prediction is, well, if I engage in that action, something good's going to happen, right? Okay, so that's what's called the teaching signal. You make predictions, you engage in an action, you have an outcome, it's better than expected, you positively reinforce that action, right? It's called instrumental learning. Okay, now the opposite could happen. You could be in a context, you can make a prediction, you can engage in an action, and the outcome's worse. Let's say you went to the other room and your pain got worse, right? Then you're going to revise your predictions, and the next time you'll be less likely to engage in that same action. So we're constantly going through life, changing context, changing, different cues are coming on, and we're making predictions about what's going to happen if I engage in this action. And then something will happen, and either you'll be correct, in which case you haven't learned anything, and you'll continue to make the same predictions in the future. That's often what happens. But when there's a surprise, when the environment changes, when the outcomes change, that's when you learn, and that's when you revise your predictions. Okay, so I want to put a little table up here because this is a very important point that I'll come back to in this talk. So we just talked about, let's say, reward. You make a prediction, let's say of a certain level of an outcome that's going to be positive. But the outcome actually is less. Let's say the reward is smaller than you expected. Then over time, or there's no reward, in which case the behavior will be extinguished. If the reward is better, then you reinforce the behavior. You're more likely to do it in the future. If the outcome is exactly the same as the prediction, then there's no change, there's no learning. You see exactly the same thing for pain, for punishment, right? You make a prediction, the pain's going to be X, and then it's even worse, right? So then that's going to lead to extinction. So note that the prediction of pain being worse, right? And the prediction and with an outcome that's worse than prediction, you extinguish. With reward, it's the opposite. If the outcome is less than predicted, you extinguish. Conversely, let's say you predict a certain level of pain and instead the pain is less, then you're going to get reinforcement. That's going to be good. That's the same as getting a reward that's greater than expected. So pain that's less than expected is identical to a reward that's more than expected, right? So let me just emphasize that. So these are equivalent for learning. And we call, relief of pain a negative reinforcer, which is the opposite of a punishment. So they're formally the same. So if you remember one thing from that slide is relief of pain is a reward that's equivalent to having an outcome, a reward that's greater than expected. And it turns out, and this is work from Frank Parecka's lab, that it's very similar circuitry that mediates the reward from pain relief as from getting a positive reward, like let's say chocolate or money. Pain relief is a reward. Okay, so just in summary, predictions inform decisions. That's why we make predictions, right? Because we want to get it right. Outcomes can change predictions, right? Expectations, and that's the subject of today's talk, expectations are the subjective correlate of predictions, right? And it turns out that expectations are powerful modulators of behavior. They not only help guide our behavior, they actually exert bidirectional control through top-down modulatory systems. Okay, so is this relevant to chronic pain? And this is a great study out of Canada, 3 different Canadian pain centers. There were over 2,000 patients, and they did complete intake interviews of the patients, and they went through a variety of factors to try to see which factors were important in predicting the outcome of treatment in a multidisciplinary pain center. And what they found was the biggest predictor of whether your pain center, your approach, whatever that approach was in these 3 different pain centers, the biggest predictor of patient outcomes was patient expectations at entry to the program. So they looked at here, variable 1 is expected relief. Here, you go down to 4, changes in pain intensity after 6 months is highly correlated with the expected pain relief here. That's 001. So expectations are incredibly important in treatment outcomes. And those are clinical studies of chronic pain. So I want to talk about a little study that John Keltner did. This is a while ago, where we were just looking at people learning about the effect of neutral sensory cues on pain intensity. So the test went as follows. So they either received 48-degree centigrade stimulus or a 47-degree centigrade stimulus. So this would have been given a pain rating of, let's say, about 8. This would have given a pain rating of around 6, right? And we preceded the high intensity stimulus with a cue. It's a red background with the words high pain, or with the low stimulus intensity, the cue was a green background on a screen, a computer screen with the words low pain, right? So over time, patients came to associate the cue with stimulus intensity. And then on random occasions, we would switch and give the high pain cue with the low intensity stimulus, or the low pain cue with the high intensity stimulus. And what we found was, not surprisingly, if there was a high pain cue and a high intensity stimulus, the average pain rating was around 8.2. If it was a low stimulus intensity and a low cue, the low pain cue, the pain rating was a little under 6. Now, if you had the high pain cue with the low pain stimulus, you got a significant increase in pain intensity. If you had the low intensity cue with the high intensity stimulus, the pain rating was significantly lower, right? So what this shows is that there's bidirectional control, that contextual cues, neutral cues, can influence pain ratings acutely. And this is an acute situation, so this has nothing to do with chronic pain. But the importance of the finding is that there are neutral cues all around. They're always present, right? You go through your life, you have these cues and they become familiar. But if you have let's say you have an ongoing nociceptive stimulus coming in, now these cues come to be associated with that pain input, right? And so it's changing the expectations. So now when you go back to the same context or the same cues are applied, that same noxious stimulus gives rise to a subjective intensity that's greater. Okay. So let's talk a little bit about placebo. This is something that everybody's familiar with, and this is one of my favorite studies from Amanzio and Benedetti. It's a long time ago. These were normal volunteers. They had a blood pressure cuff put on their arm, pumped up above systolic, right? And then they were exercising. And the idea was, how long could they exercise under ischemic conditions before they had to stop due to the pain, right? So the control on day 1, they didn't do anything. And the average duration that people could tolerate was 15 minutes of this exercise. However, on day 2, they started an infusion. It was saline, so it wasn't anything. And they said, this is a powerful painkiller. And on average, there was a significant but relatively small increase in people's ability to tolerate this ischemic muscle situation. And then they go back on day 3 just to see that it's reversible. There was no infusion, and they're back to where they were before. Now, here's the beautiful part of this experiment. So it's very similar setup, except that on day 2 and day 3, they actually got an infusion of morphine, right? And then after having those infusions of morphine, they're given a saline infusion on day 4. So day 4 is equivalent to day 2 prior to the morphine conditioning. And you can see that the conditioning greatly enhances the placebo effect, right? So you can train the brain and improve people's, confidence in their expectation of relief, and then they get a bigger placebo effect, right? So the placebo effect is not a standard level of relief, or it's something that can change in people based on experience. So here's a very powerful expectation effect. I love this study. Okay, so what is this all about? What's the biological value of being able to modulate pain intensity? Why would you want to do that? Obviously, this is a critical signal. You need it for tissue protection. Why would you want to suppress it is there any biological value in suppressing pain? And the answer is, of course, yes. And that can only be understood in the context of conflicts with competing motivations that are also essential for survival. So again, we're getting back to the biology and the evolutionary meaning, of modulation, of pain modulation, of the variability of the experience of pain, and why under certain circumstances, it actually might be to your advantage to be able to ignore and suppress the pain signal, or at least suppress the responses to the pain signal, right? So let's say you're hungry, you haven't eaten in days, right? And you're gonna have to do something that really hurts, to get dinner. Maybe it's to, endure a few bites from whatever prey that you're trying to capture. Anyway, you get the point. So this really, I think the systematic study of this began early in the previous century. Pavlov did this famous experiment with dogs. I'll just read the slide. He said, when a noxious stimulus is rapidly and repeatedly followed by feeding, all signs of pain are lost and replaced by approach and salivation. In other words, because the noxious stimulus is a sensation, it can be a cue to a rewarding situation, right? So yeah, it's painful, but it's predicting a pleasurable outcome, a reward. In this case, it's food, which is very important. And it appears as though the animals are not experiencing any pain. In fact, they might be experiencing, paradoxically, pleasure from this noxious stimulus, which is a cue to reward. And a little bit later study, a little bit more systematic than Pavlov, this came out of Albert Hertz's lab in Germany. And this goes, I love this experiment because it's so cheap, right? They just had a hot plate, right? They put the rat on the hot plate. They didn't turn the hot plate on. They just put the rat on a hot plate for a few times over a period of weeks. And then they would feed them either their regular lab chow, in which case there wasn't much of an effect. Then after they had the animal on the hot plate, never turned it on, was fed on the hot plate, the regular diet. Then they turned on the hot plate and measured the latency for the rat to get off, right? And it was around 5 seconds. Now, in another group of rats, they fed them chocolate-covered graham crackers or cookies. Same situation, but then when they turned the hot plate on after the training, the rats were able to stay on significantly longer, right? So their expectation of a reward was able to turn on their pain suppression system so they could wait a little bit longer to get that highly desired reward, right? So here we're getting at this place where pain and reward are opposite hedonic values, but they're always interacting based on predictions, right? You make a prediction of pain, you're going to have pain, rewards become less valuable. You make a prediction of a reward, pain becomes less painful, okay? Very simple. There seem to be, this type of work's been replicated. I think that's what's going on with placebo. I think it's an expectation of a reward, i.e. the relief of pain. How does this happen? Well, in addition to this bottom-up pain sensory system, there's also a top-down pain modulatory system. This is something that I worked on for years with Alan Bassbaum, Mary Heinricher, and other people in the lab. This was our big interest. How did this top-down system work? Runs from cortex, some of the same areas of the cortex that are activated by the noxious stimulus. There's some new parts to this system that aren't shown on the slide, including the parabrachial nucleus. This area, the periaqueductal gray, is in the midbrain. It receives cortical inputs. It projects to an area in the medulla, and in the medullary area has neurons that project directly to the spinal cord it's not just to the spinal cord, but they project specifically to those parts of the spinal cord that receive input from the small diameter nociceptive fibers and contain second and third order neurons, which transmit the pain message. So you have this whole brain system that's been activated by noxious stimuli or external neutral cues, feeding down through the midbrain medulla and controlling the pain message as it enters the spinal cord. So this information about nociception doesn't get through. It doesn't enter any part of the nervous system. It even suppresses withdrawal reflexes when the system is fully activated. Does this happen in people? And it was very gratifying to me to see these publications about a decade ago out of Christian Buchel's lab in Hamburg, where they were looking at people they gave that had a pain state, where they were given a placebo. Some of them responded, and they correlated the relief that people got from the placebo to blood flow changes in different areas. And these areas that they found correlated to areas in the brain of rodents that are part of this pain modulatory system that we talked about in the previous slide. Even incredibly, they were able to functionally image the spinal cord. So here's the spinal cord, down is dorsal here. This little blip is activity in the dorsal horn produced by a noxious stimulus in the periphery. Pretty incredible. And then that activity went away with giving a placebo. So if you got placebo relief, not only did you activate these centers in the brain that are part of the modulatory system, you could also demonstrate that they're having an inhibitory effect at the level of the spinal cord. So this is a pretty good confirmation of the idea that placebo or the expectation of relief engages this top-down system. It's always incredibly gratifying when you can see that work that you've done looking at neurons in the central nervous system of animals in preclinical studies is validated in studies of humans who are able to tell you, yeah, my pain is less, and you can correlate the changes in activity of different areas, in this case, the modulatory system and the spinal cord, with the reduction in the pain report. So this is pretty incredibly gratifying work from Buchel. I'll talk a little bit about the mu-opioid receptor. Definitely my favorite molecule in the brain. This was cloned in 1982. This is a review from Brigitte Kiefer, who's in France. She was the first to make a mouse with the gene deleted for the mu-opioid receptor. And once a mouse that doesn't have the mu-opioid receptor functioning gene, you can give them a ton of morphine, no analgesia, no reward, no respiratory depression, no nothing. So you can completely explain the effect of morphine and drugs like that on the basis of their action at this mu-opioid receptor. So it's pretty critical, and it's a very potent effect, as you know, if you've ever had or prescribed an opioid. And Brian Kavilka, actually at Stanford a decade ago, he got the crystal structure of the mu-opioid receptor. So and that can be used to help design new and better opioids. So it isn't enough to have a receptor, you have to have some kind of chemical that acts at that receptor, right? And it turns out that in 1975, Hughes and Kosterlitz reported that they had extracted a couple of small peptides, and this is methionine and leucine and keflin, from the brain of a pig that had the properties of morphine, right? I mean, you got these peptides, you could put them on these experimental tissues, and they had the same effect of morphine, and you could block them with an opioid selective antagonist, naloxone, right? So that was a big event. And then once you've got those peptides, you can make antibodies and you can trace their location in the central nervous system of animals and people for that matter. And it turns out that, guess what? Every site along this top-down pain modulatory pathway, including in the superficial dorsal horn, have endogenous opioids. They also all express the mu opioid receptor, so you can inject morphine into any of these sites and get pain relief. Anyone that's done intrathecal opioids, you know that they can act directly on the spinal cord to relieve pain. So this is cool. So you've got endogenous opioids, you've got the mu receptor, they're in the central nervous system. We know that you've got top-down modulatory circuit, and here it is, the endogenous opioids are present in this top-down system. Do they do anything meaningful in patients? And let's go back to this beautiful study that I love from Benedetti's lab, where they basically did the same, procedure here, and then instead of giving saline on day 4, they gave naloxone on day 4, and they got no placebo effect, right? And so this kind of validated some work that John Levine and I did back in the 70s, where we would block the placebo effect with naloxone. So it looks like the expectation of, let's say, the reward of pain relief led to the release of an endogenous opioid acting at the same receptors that morphine acts at. So this is kind of cool because, right, you've trained the person, these are people, with morphine, and then when you give them what looks like morphine, they release something in their own brain that acts at the same receptor that morphine's acting at, right? So you're kind of mimicking what happens pharmacologically through expectation, right? Again, confirming the power of expectation to modulate pain. I keep coming back to that because I think if there's one message from this talk, it's that. Okay, so remember the earlier study that I talked about with John Keltner, where we had these neutral cues. What that showed was that this expectation effect is bidirectional. So you don't just have top-down inhibition, you have top-down facilitation of pain, right? And it turns out that if you look at the individual neurons in this pain modulatory pathway, there are two types of neurons that have opposite firing patterns. And through a variety of experiments, we were able to show that one of these cells, which seems to shut down just before the animal withdraws from pain, we called that the off cell because they'd have to shut off to allow the rat to escape from pain, right? So we call that the off cell. And you can demonstrate that that actually has an inhibitory effect on pain. You have to shut it off in order to respond, right? Then the other type of cell in the same area, which we call the on cell, shows a short burst just before the animal withdraws, right? So, and we had a set of experiments to show that that cell, if you selectively activate that cell, you can get pain facilitation. So pain, these are cells that are present in the PHE and in the medulla, including those cells that project directly to the dorsal horn. Bidirectional control, right? So here's a cartoon illustrating that. You have the nociceptive input coming to the transmission cell, and it can either be inhibited or excited. So the message that gets to the brain is an additive function of your input and your top-down, in some cases, learned expectations activate either a facilitating neuron, the on cell, or an inhibiting neuron, right? So if you have a given input, let's say of 1, and you have a descending input of 1, then your output is 2. If you have an input of 1 and you have an inhibition of 1, then the activation of this T cell is 0. Very simple. And really beautiful, robust effects of morphine. If you give morphine to the systemically in this case, or you can give it locally, so the morphine's right next to the neurons that you're recording from, that's shown here. Let's you give morphine and then you get an increase in activity of the off cell. So morphine increases the activity of the pain inhibitory cell, and then it inhibits these pain facilitating neurons. So it has a double effect, right? It's promoting inhibition and it's turning off facilitation. That's another reason that opioids are so powerful. Okay, so you have bidirectional control. Opioids have, opposite effects on the inhibitory and excitatory cell. All right, so let's talk about what this is all about. Why do you have this bidirectional control? What is it doing? The conflicting goals demand a decision, right? Okay, so you have to make a decision. You're going to do a cost-benefit computation that precedes your action. You have a noxious stimulus. You have these conflicting drives. You have a cost-benefit computation. You make a decision. And then if it's to ignore the pain, you have an opioid release, you increase your off cells, and you have a decrease in pain intensity. Conversely, if the decision is, I got to get out of here, I have to respond, then you got your on cells facilitating, you have an increase in pain intensity. So let's get to where in the brain this decision's being made. And I talked about the nucleus accumbens. It receives a major dopaminergic projection from the midbrain. This was discovered. Animals will press a lever to receive electrical stimulation. So it was originally thought of as a reward pathway. And in fact, it's activated by drugs of abuse. And that's all very confirmatory. If you microinject an opioid into the nucleus accumbens, it promotes feeding and it suppresses pain. So it does what it should do if this nucleus accumbens is involved in decisions. Turns out in the human, there's a high density of mu receptor in the nucleus accumbens. So this is the same area that is different in the chronic pain patients from the patients from normal volunteers, right? A lot of mu receptor in the nucleus accumbens, and using a similar C11, this is a it's a carbon molecule with a half-life of 20 minutes. So you can give it twice, you give it once, you see the binding, you give let's say a placebo, the binding goes down, that means an endogenous opioid's been released. This is showing that with placebo analgesia, there's release of an endogenous opioid in this decision circuitry. But the decision circuitry is also activated by pain predictive cues as well as reward predictive cues. So I'm not going to go into detail about that study. Let's just say that the nucleus accumbens shows an increase in activity with pain predictive cues and with reward predictive cues. Let me switch over. And it turns out that the nucleus accumbens itself has 2 types of neurons in it. These are different dopamine receptors. D1, these neurons tend to be rewarding when they're activated. The D2s tend to be aversive and pain facilitating when they're activated. Okay, so the pain activates the D2s and you get hyperalgesia. This is work from the Ren lab in 2016. Morphine activates the D1 cells. This is Annexon's work again a few years back. It's rewarding. You get and if you activate these cells, you get an analgesic effect. And that's a very recent paper out of Sato's work in Japan. So you got the nucleus accumbens bidirectional output. So here's the model. Prefrontal cortex, you get a pain predictive cue, or you get a reward predictive cue. The pain predictive cue facilitates pain. The reward predictive cue inhibits pain. Now, we're not quite clear on how the nucleus accumbens talks to the pain modulatory circuit, but it seems like it must. Okay, so in summary, pain occurs in the context of conflicting motivations. That's the key sentence. because you have conflicting motivations, you have to do a cost-benefit analysis. In order to do that analysis, you have to make predictions. Predictions are essential for optimizing decisions, based on reward approach or pain avoidance. The subjective correlate of that is expectation. As I said, the descending control is bidirectional. I can go over that. So the key point clinically is that you can learn to feel, or you can learn to ignore pain. Pain predictive cues increase pain. Pain itself is pain predictive. Expectation of future pain, goes down with early effective intervention. The expectation of pain increases with each ineffective intervention. So you can either have a vicious cycle where you have pain predictive cues, the pain doesn't, continues, and you just reinforce the expectation that you're going to have pain. That makes the pain more intense, and then the expectation goes up, right? Or you can have, relief and a virtuous cycle, and then pain gradually fades. For some patients, an improvement has to involve changing expectations. So this is sort of the slide that you should remember. Pain can worsen over time with no change in nociceptive input. The expectation of future pain increases. That is the vicious cycle. This is a learned CNS dysfunction. What can you do clinically? Assess patient expectations. Really, really important. People can hope that you're going to help them, but expect that you won't. And it's important to counter this unrealistic pessimism with facts. Set realistic goals, create a therapeutic alliance. And finally, I'd like to say that optimism is useful and ethical, and scientific progress, at least in my mind, supports an optimistic attitude for many of our patients with chronic pain. Patient-centered research is the key. So keep it up. Good luck. And with that, I'd like to thank Beth, Ashley, and SPRIL for inviting me, and all of my colleagues. And I'd be glad to take questions.
Oh, thank you so much, Howard, for a terrific talk. You and I have published on the topic of opioid tapering, and in particular forced opioid tapering. And, without having to give a completely separate lecture, are there some key takeaways that can inform why the practice of forced opioid tapering is particularly detrimental for patients?
Well, thanks for asking. I wanted to talk about that, but I knew I wasn't going to have time. And I think that the most important data related to that is the research that's been done on pain controllability and pain intensity. So there's some beautiful studies, again, with normal volunteers, whether, the same pain intensity, whether they can control it or whether it's completely investigator control. And there's a highly significant correlation with people's subjective experience of uncontrollability and how intense the pain is. So, unless you're doing the taper with the full consent and participation of the patient as agent, you're almost doomed to fail.
Right. Right. Thank you. Thank you for that. We had a lot of great questions. Is pain a sensation or a perception? And what are your thoughts on cognitive penetrability and pain?
Well, certainly pain is a sensation. It's also a perception. It's also a motivation, right? And it has multiple sides to it. One of them is actually the quality and intensity. This seems to be computed in somatosensory cortex, but there's also the unpleasantness and aversiveness, and I call this the motivational side. That seems to be a separate parallel circuitry. So you can, impair people's ability to locate and assess the pain, and it still retains its aversiveness. Conversely, you can block the aversiveness and still, oh yeah, that's pain in my finger. I give it a 10, but it doesn't bother me. So I don't know if that fully answers the question, but.
Is it possible to experience pain without nociception?
That's a great question. And I think it's related to, though there's two ways to approach that. The first one is, can you experience pain if there's no activation of nociceptors? And I would say absolutely yes. And the reason we know that is that there are places in the brain that neurosurgeons have accessed, and this would be in the ventral posterior lateral thalamus. So you can put an electrode down there and stimulate. There's no nociceptive input, right? But the patient says, oh yeah, I feel a burning, in my foot, right? So in terms of, you don't need a nociceptive input to feel pain, right? So, and then I guess the other side of it would be nociception or sensation as opposed to the aversiveness. They're separate. So you could presumably activate the aversive circuit, which includes anterior cingulate in particular, right? And get somebody to say, oh yeah, that really feels terrible. I don't know whether I'd call it pain or not.
Thank you. How does anxiety fit into the expectation model? Okay,
so this is a really good question. Again, these are all great questions. So there's been a lot of research on that actually, and anxiety, if you're anxious about the pain, then the pain is actually felt as more intense. If you're anxious about something other than the pain, then it actually can have an analgesic effect, right? So it's where the anxiety is focused that seems to be important.
Fascinating. This is from Sean Mackey. Terrific talk, Howard. Why do you think that motivation did not make its way into either the original IASP definition of pain or the revised definition when it is so fundamental?
Yeah, so, well, I have a philosophical answer to that, and that is that by and large, over the last 30 to 40 years, pain research has been driven by psychophysics, right? As opposed to biology. In biology, we're always trying to figure out, well, what's it for? What's it doing? It's interesting that Pat Wall himself said pain has to be thought of in the context of an action decision, right? But the people that were doing the taxonomy, that were making up the definition, were not biologists, right? There's nothing wrong with their definition. It's just incomplete.
Thank you. This is from—
What great questions. We should just go on and on. I don't want to stop.
I know. When we're past the top of the hour, let's do one more.
Okay.
This is from Dennis Given. It would seem that presenting outcome data to patients supporting the effectiveness of a multidisciplinary treatment program would increase the likelihood of those patients benefiting from the treatment.
Yeah, you would think so. But the only way you're going to find out is by actually doing it.
Studying it, yeah.
Well, you have to study it because, if people are skeptical about, pain clinics in general, they may get the information, but they may not believe it. That's why I say it's, people would come into my office when I was seeing patients and they'd say, look, you're my last hope. I've seen everybody, I've done everything, I've had every treatment, and I've run out of options. And I'd say, well, do you think I can help you? And they'd say, not really. So you're already starting off in the hole. And that's why I think that it's really important. It's an educational process to try to, get away from the unsupported pessimism, right? Of catastrophizing.
Well,
know that it's true because it's kind of core to your approach.
I think it really nicely illustrates the detriment that is caused by waiting until all medical treatments have failed to integrate in pain psychology and to then refer to a psychologist. It's such a disservice to patients. It's such a disservice to the profession that could potentially help a person, but what is being encoded is failure. And then, oh, the physician's giving up on me now. They're sending me out to the psychologist. My pain isn't real. It's all psychological. And nothing could be further from the truth. So it's actually, the deficiencies of how we are integrating or not integrating our treatments, the order in which we approach them, the messages that we give. We've really failed patients in so many ways.
I could not agree with you more. That's exactly right. It's backwards.
It's backwards. It's backwards. And, and your research nicely shows that. And a lot of us here, even on this webinar, are on a mission to change that. Howard, thank you so much for your generosity and your time in sharing your life's work and your wisdom with us.